So, hello students, this is Dr. Ravi Sani, and today we are going to discuss about the active transport. So, that is really important topic, which is usually asked in various exams. So, that is active transport. As its name suggests, active transport. So, there is a utilization of energy. So, in the transport process, which utilizes energy, and definitely if it is using energy, it is going to be against the gradient. So this one is going against the gradient, and there is a utilization of energy. So this is the key conditions in the active transport. There should be utilization of energy, and the movement of the ions should be against the gradient. Basically, nature tries to maintain inequality between the intracellular and extracellular compartments. So these special transport mechanisms helps in maintaining inequality, and this active transport helps to maintain that particular condition. So the active transport basically could be divided into two types. The first one is the primary active transport, and the second one is the secondary active transport. Now we are going to discuss about the basic differences among these two terms. Now come to the primary active transport. It is a transport mechanism which directly utilizes metabolic energy for the transport, and that mechanism could be operated by the ion pumps or ATPs. So there is a direct utilization of energy through which an ion is moving against the gradient, against the concentration gradient. These are the various examples of primary active transports. Sodium, potassium, ATPs, it is a very common example. Calcium ATPs, hydrogen potassium ATPs, hydrogen ATPs, mitochondrial ATP synthesis, CFTR protein, MDR1 protein. These are the various examples which shows primary active transports. The most important and the common example of primary active transport is the sodium, potassium ATPs. So basically, it is an antiport that pumps potassium into the cell and sodium out of the cell. Antiport means there is a movement of these two respective ions, the sodium and potassium, but both of them are moving in opposite direction. So that is antiport. By the help of this mnemonic, we could easily remember the movement of these ions. Nokia. So there is a movement of sodium outside, and potassium is moving inside the cell. So this antiport system is primarily responsible for maintaining the high potassium and low sodium concentration inside the cell. So basically there is a movement of 3 sodium ions outside the membrane, and 2 potassium are moving inside the cell. So there will be a increased concentration of sodium outside the cell. There will be increased potassium concentration inside the cell. Now come to the importance of that particular sodium potassium ATPs. So basically it is moving ions against the concentration gradient. So by this movement, by the movement of these ions, an electrical potential is generated across the membrane. which is called resting membrane potential. So sodium potassium ATPs helps in generation of electrical potential difference across the membrane. Usually we call it resting membrane potential, and that resting membrane potential is very, very important in transferring signals, specifically nerve and muscle fiber. In further classes, when we'll deal about the nerve physiology or muscle physiology, where we are going to discuss about this resting membrane potential in detailed manner. So this sodium potassium ATPs helps in generation of electrical potential difference across the membrane. Usually we call it resting membrane potential, and this resting membrane potential is responsible for the transportation of signals or transporting signals. So we discuss about the primary active transport. Now it is secondary active transport. So definitely energy is also going to be used in that particular transport as well, but there will be a movement of two ions. The first one is called driving ion, and the second one is the driven molecule. So there will be two molecules. The first one is the driving ion, and the second one is the driven molecule. So commonly, sodium would be the driver solute for most of these mechanisms, and the energy created by the sodium gradient will help in transport of other solutes. So in primary active transport, basically energy was used directly and ions were moving across the membrane. It could be in the same direction or it could be in the opposite direction. In secondary active transport, there is a driving ion, there is a movement of driving ion, and by generation of moving by the movement of this driving ion, energy is being generated, and that particular energy is used to move the another ion across the membrane. Similarly, here also the direction of the ions could be in opposite direction or in same direction. If it is in same direction, it is called symport, and if it is in opposite direction, then it is called antiport. In primary active transport, energy was used directly. In secondary active transport, energy is being generated by the movement of driver solute for transportation of another molecule. So basically, the transport is depend on the primary active transport of sodium. So in the secondary active transport, basically secondary active transport is dependent on the primary active transport of sodium by the sodium potassium pump. That's why it is called secondary active transport. In primary active transport, the direction of the respective ions could be in same direction. It is called symport. It could be in opposite direction. That would be called antiport. Similarly, in secondary active transport, also the direction of the movement of these ions could be in the same direction or could be in the opposite direction. The common example of secondary active transport is the sodium glucose co-transport. So in PCT, there is that is proximal convoluted tubule, which is the part of nephron where reabsorption of glucose is done by this secondary active transport. Here we calling it sodium glucose co-transport. The word symport refers to direction of these two ions, sodium and glucose, in the same. So in secondary active transport, basically sodium ions when sodium ions are transported out of the cell by the primary active transport, as we studied in the sodium potassium ATPs, that is the example of primary active transport, sodium was moving outside the cell. So there is a development of concentration gradient because higher concentration of sodium is being developed outside the cell by the help of sodium potassium ATPs. So there will be a generation of large concentration gradient across the membrane. So sodium will always try or will have a tendency to move inside the cell because there is a high concentration outside the cell because of the sodium potassium ATPs. When sodium was moving out of the cell, and there is a less concentration inside the cell. So this movement of sodium by the help of sodium potassium ATPs will cause higher concentration outside the cell and lower concentration inside the cell of the sodium. So sodium will always have a tendency to move inside the cell. So this gradient will help movement of other ions. So that's why it is called co-transport. If it is in same direction, and then it will be called counter transport if the movement of these ions are in opposite direction, that is symport or antiport. So under appropriate condition, the diffusion of energy of sodium can pull other substances along with the sodium through the membrane. So by the help of primary active transport, sodium was moved out of the cell, concentration gradient was generated, and due to this concentration gradient, sodium will always have a tendency to move inside the cell, and this movement of sodium from outside to inside is responsible for the movement of another ion, and that could be in similar direction or could be in opposite direction. If it is in same direction, then it is called co-transport or symport, and if it is in opposite direction, then it is called exchange, counter transport, or antiport. This whole mechanism, basically there is a carrier protein which is responsible for the movement of these ions. The carrier protein has an attachment point for both the sodium ion and the substance which is co-transported with the sodium. So there is a carrier protein which has two attachment points, one for the sodium and one for the another substance that is being co-transport. In this case, there will be a movement in opposite direction. And in this case, these are the two attachment points that could be in the same direction. These are the attachment points in antiport, and these are the attachment points in symport. So in counter transport or in antiport, sodium ions have a tendency to diffuse interior of the cell because of their large concentration gradient. That concentration gradient is generated because because of the primary active transport which moved sodium out of the cell by using energy. So there is a higher concentration outside the cell and lower concentration inside the cell, and this higher concentration of sodium will always have a tendency to move inside the cell. When this movement of sodium will occur, it will also help in movement of the other substance. It could be inside the cell or it could be outside the cell. So in sodium glucose co-transport, there is a transport carrier protein which has two binding sites. This is the transport carrier protein. It has two binding site. One is for sodium binding site and the other one is glucose binding sites. The concentration of sodium ion is very high on the outside and very low inside, which provides energy for the transport. Basically, why this concentration gradient is generated? Because in primary active transport, by the help of sodium potassium pump, sodium was moved against the concentration gradient, so there is an increased concentration outside the cell. So due to this, there is a generation of gradient. This gradient is responsible for the movement of these sodium ions. So in this carrier protein, there are two binding sites. One is for sodium, and the another one is another one is for glucose. So sodium glucose transport occurs in the same direction. It is called symport or co-transport. So when this sodium will move inside the cell, there will be generation of energy, or this movement of sodium will help in movement of glucose as well. Both ions are moving in the same direction, and it is called co-transport or symport. There is a special property of the transport protein that a conformational change allows sodium movement to the interior, and this movement of sodium will not occur until the second molecule or second ion or chemical compound which is going to be transported will not get attached until. So in this sodium glucose co-transport, sodium there will be no movement of sodium until glucose is also got attached with that particular transport protein. So when these two ions or molecules got attached with that transport protein, then will be movement of the ions or chemical compounds take place, and that will cause the movement of sodium and glucose in the same direction or inside the cell. It is called sodium glucose co-transport. It is in the same direction. It is also called symport. Now come to the other examples of sodium co-transport or symport, because the sodium and the other molecule which is going to be transported are moving in the same direction. So it is co-transport or symport. Examples are, the common example is the sodium glucose co-transport. Both are moving in the same direction inside the cell. Sodium phosphate co-transport. Sodium is moving inside the cell with phosphate. Sodium iodide co-transport. Sodium potassium chloride co-transport. Sodium chloride co-transport. Potassium chloride co-transport. They are moving outside the cell, but both the molecules have same direction. These are some other examples of sodium co-transport. So in secondary active transport, the another example is the counter transport or exchange or antiport. Till now we have studied primary active transport, in which energy was specifically used for the transport of particular molecule like sodium potassium ATPs. ATP energy in ATP form was used in the movement of sodium and potassium. Sodium was moving outside the cell, potassium was moving inside the cell, and it was responsible for the generation of potential difference, which we usually called it resting membrane potential. After that, we discuss about the secondary active transport. In the secondary active transport, energy was basically energy was used in transport of some other molecules like sodium potassium ATPs was responsible for the movement of sodium outside the cell. The sodium was having a higher concentration outside the cell than the inside of the cell. So there is a generation of concentration gradient. So sodium will always have a tendency to move inside the cell. So when this sodium is moving inside the cell, it will also responsible for the movement of another ion, and it could be in same direction, then it is called co-transport or symport. And in this example, the transport of another molecule is in opposite direction, then it is called counter transport exchange or antiport. The examples are sodium calcium exchanger. Here we can see there is a higher concentration of sodium outside the cell because of the sodium potassium ATPs. So there is a generation of concentration gradient. Sodium is moving inside the cell, but the calcium is moving outside the cell. So there is an opposite direction. It is called uh, counter transport, counter secondary active transport, or exchange or antipode. The another examples are sodium hydrogen exchanger or chloride bicarbonate exchanger. There is a opposite direction. So we could also have a basic idea about various carrier proteins. There is a uniport carrier protein, symport carrier protein, and antiport carrier protein. So uniport carrier protein basically it transports only one substance, as its name suggests, uniport. So it transports only one substance. It could be sodium, potassium, or some other ion. In case of symport carrier proteins, it transport two or more substances from one side of the membrane to the other, but in same. It could be movement of two ions, or it could be movement of more than two ions, but in same direction. Then it is called symport. A common example is a sodium glucose co-transport. The protein involved in the transport of sodium and glucose is having a type of symport carrier protein. In antiport carrier protein, there will be a opposite direction of the movement of the respective ions. The common examples are the sodium potassium pump that is responsible for the primary active transport, sodium potassium exchanger. In both the cases, there is movement of ions in opposite direction. So in this image, we could have a rough idea about the membrane transport processes. So we have simple diffusion, we have passive diffusion, we have facilitated diffusion. In passive transport, so broadly, we can divide the transport into active and passive. In passive, there will be no requirement of energy. In active transport, definitely there will be a requirement of energy. The active transport could be divided into primary or secondary. In primary, energy was specifically would be used for the movement of that particular ion. The common example are the sodium potassium ATPase. In secondary active transport, energy is used definitely, but in an indirect way, because some other ion was moving, and by the movement of this the other ion is responsible for the movement of respective ion. The common example are the sodium glucose co-transport. So in secondary active transport, when sodium is moving across the concentration gradient, it is helping to movement of some other ion. It could be in same direction, then it is called symport, and if it is in opposite direction, then it is called antiport. In passive transport, there will be no requirement of energy. It could be simple diffusion, passive diffusion, or facilitated diffusion. In facilitated diffusion, there will be a use or role of specific protein which helps in movement of the ions.